So after discussing the structure of the eyeball in a very short aspect, we will move towards the histology of the cornea. So the thickness of the cornea is approximately 1 millimeter, which is having the convexity in the forward aspect, as well as cornea is supplied by the numerous nerves. Now, according to the structure, the cornea is divided into the 5 layers from anterior to posterior or outer to inner aspect. First layer here you can see that is known as the corneal epithelium. After the epithelium, the second layer is known as the anterior limiting membrane or the Bowman's membrane. Third layer, that is approximately occupying the 90% of the thickness of the cornea, that is known as the corneal stroma. Fourth layer, that is again one membrane, that is known as the Descemet membrane or also the posterior limiting membrane. And last one, fifth one, that is known as the corneal endothelium. So these are the 5 layers from anterior to posterior aspect. First we will discuss the corneal epithelium. So the epithelium here, that is of the non-keratinized stratified squamous type of the epithelium, which is having the rapid regeneration property, approximately 7 days required to regenerate. And within this epithelium, the deepest layer of the epithelium, that is having the low columnar types of the cell having the round nuclei, and superficial layer, that is having the disc shaped flat nuclei with the pycnotic nuclei, you can also say pycnotic nuclei. And during the migration process, the cells are dividing rapidly from the deeper layer and migrated towards the superficial aspect. During that process, they almost lost the organelles. Now, stem cells of this corneal epithelium, they are present in the limbus region, corneoscleral junction. Now, corneal epithelium is also supplied with the free nerve endings, that is making cornea extremely sensitive for the touch, and ophthalmic division of the trigeminal nerve, that is supplying this area. Now, the superficial layer of the corneal epithelium, that is also showing the microvilli onto the surface. Now the second layer, that is lying onto the inner aspect of the corneal epithelium, this layer number 2 shown in here, that is known as the Bowman's membrane or the anterior limiting membrane or the anterior basement membrane. Now this layer is homogeneous in nature, okay, having the homogeneous laminar arrangement, and there is an arrangement of the collagen fibrils, that is also randomly arranged collagen fibrils, and this layer is acting as a barrier to prevent the infection from the outer aspect, as well as that is going to support the corneal epithelium. Now this layer is synthesized by both corneal epithelium as well as the corneal stroma. Now if there is a damage to this membrane, that is resulting in a impaired vision due to the development of the opaque scar. The third layer, that is occupying approximately the 90% of the thickness of the cornea, that is known as the corneal stroma or the substantia propria. Now there is an arrangement of the lamellae within this layer, and that is containing the type 1 collagen fiber as well as the ground substance. Now within each lamellae there is an arrangement of the collagen fibers that is parallel to each other within each lamellae. Now within adjacent lamellae, if we trace the direction of the collagen fibers, that is perpendicular to each other in adjacent lamellae. And this type of arrangement, regular arrangement, that is termed as the orthogonal array. So this type of orthogonal array arrangement, that is giving transparent stroma and transparent nature of the cornea. Now between the lamellae and ground substance, there is a presence of the fibroblast also. Ground substance is mainly made up of the proteoglycans, and fibroblasts, the shape of the that is something looking like star, and these fibroblasts are also known as the keratocytes or the corneal corpuscles. Okay, and we have already discussed that canal of Schumann that is present at the corneoscleral junction, that is present here within the corneal stroma, that is helpful to drain out the excess fluid of aqueous humor from the anterior chamber into the venous system. Now the fourth layer that is also lying here, that is known as the basement membrane or the posterior basement membrane or posterior limiting membrane. It is acellular as well as the homogeneous in nature, and this is particularly termed as the true basement membrane. And there is a interwoven meshwork of the collagen fibrils that is found in this layer, and this membrane is the product of the corneal endothelium that is lying here. So we can say that is completely true basement membrane. Now another term that you have to understand, that is known as the pectinate ligament in concern with this basement membrane. So the peripheral fibers from this basement membrane, they are extending beneath the sclera to form the meshwork. Okay, and this is known as the pectinate ligament. So remember, the peripheral fibers from the basement membranes extend beneath the sclera to form the meshwork, and that is known as the pectinate ligament. Now this ligament is helping to maintain the corneal curvature. Okay, and here within this basement membrane, regeneration is possible if there is a damage, while in the Bowman's membrane, regeneration is not possible once there is a damage that is healed by the opaque scar that results in impaired vision. Now the last layer, that is the corneal endothelium, number 5th shown in this figure, that is the most posterior structure, and that is mainly made up of the simple squamous as well as the low cuboidal type of the cells, and this layer is lying in close contact with the aqueous humor in the anterior chamber, and cells are connected or in close contact with each other by the means of the zonula adherens as well as the zonula occludens and the desmosomes. Now this layer is mainly helpful to maintain the water balance in the corneal stroma. So it is pumping out the extra water that is present in the stroma and maintain the transparency of the cornea. Now the last point, that is the corneal transplantation. That is most successful transplantation, and the reason for its successful nature is due to the avascularity of the cornea. Okay, so because of the avascular nature of the cornea, corneal transplantation is the most successful of the all transplantation. So after discussing the histology of the cornea, we will discuss the histology of the sclera. So the thickness of the sclera is approximately 0.6 to 1 mm, and it's one kind of the external thick white opaque fiber cover of the posterior 5/6 of the eyeball, which is consist of the four elements. Number one, that is the bundle of the collagen fibers. Number two, that is the few elastic fibers. Number three, that is the ground substance. And number four, that is the scattered fibroblast. Now, the reason for white color or the opacity of the sclera is that the arrangement of the collagen fibers is irregular. So it is making the sclera white or opaque in nature. Now this is the schematic diagram to show you the structures of the sclera. So it is divided basically into the three portions. The outer portion, that is known as the episclera, middle portion, that is known as the Tennant's capsule or the substantia propria, and most inner portion that is known as the suprachoroid lamina or the lamina fusca. So within the episclera layer, you will find the loose connective tissue, and within the loose connective tissue, you will see the arrangement of the blood vessels as well as the nerve. So this layer is pierced by the numerous blood vessels as well as the nerve. Out of that, most important nerve that is known as the optic nerve. Now the middle layer that is known as the Tennant's capsule or the substantia propria, which is mainly made up of the dense network of the collagen fibers. But here the arrangement of the fibers that is irregular. So this type of the arrangement that is making sclera somewhat opaque as well as the white in color. And the last layer that is known as the suprachoroid lamina or lamina fusca, and that is made up of the dense delicate connective tissue fibers. Okay, so that's something regarding the histological aspect of the